Welcome all. Do you have immune cells that thought SARS-CoV-2 infection? Hey everybody, I am Edwin Williams. My endeavor is to explain complex scientific research in very simple, easily understandable way within a few minutes. Recent research explains why some people never test positive for COVID-19 in spite of having close contacts with COVID-19 patients. Some thought that it was of their blood group. A group of researchers from UK has evidence that this resistance comes from memory T cells, a type of lymphocytes in their WBCs. Let us further examine. This group never get COVID-19 infections even if they are not vaccinated, not produce antibodies against it. Their immune cells might abort SARS-CoV-2 and they will be negative for polymerase chain reaction or PCR test. This is because their bodies are able to quickly clear a nascent SARS-CoV-2 virus from their bodies even without producing antibodies against them. This study by Dr. Leo Sadling et al. Nature, November 10, 2021 suggests that the resistance in these people comes from memory T cells, a type of lymphocytes in the white blood cells of the immune system. It plays a central role in the adaptive immune response of acquired immune system composed of specialized systemic cells and processes which eliminate the pathogens, that is disease producing organisms, or prevent their growth. The study conducted on 60 UK healthcare workers who were high-risk group as they work in hospitals never tested positive for COVID-19 nor produced any antibodies for the virus for the 4 month period after enrolling in the study. The researchers also noticed that the T cells in 20 of these seronegative participants, that is, the blood serum was negative for COVID-19, the T cells multiplied, indicating that the immune system might be gearing up to fight an infection. Out of these 20, 19 also had an increased level of immune system protein IF527, that is, interferon alpha inducible protein 27, which, according to the authors, might be a biomarker, a biological, usually protein substance, which may be the earliest detectable sign of presence or absence of a disease or condition. If this assumption is tested to be true, COVID-19 infections can be nipped off in the bud. This enormously high production of IFI27 is an evidence of an abortive infection, that is, the virus already entered the body but failed to bloom into an infection. The study found further evidence that their T cells disabled a cluster of viral proteins known as the replication transcription complex, which help the virus to multiply. They found that a higher proportion of T cells recognize this particular complex in the blood of seronegative participants than did in the blood of healthcare workers who got COVID-19. It might mean that the T cells of healthcare workers who were COVID-19 negative could recognize SARS-CoV-2 and more strongly recognize the replication transcription complex and so prevented the SARS-CoV-2 by destroying them before causing the disease. The study assumes that the T cells in seronegative participants could have been generated in their bodies by other coronavirus infections that caused common cold in them earlier. There is no direct evidence as to what point of time these T cells originated in this group. The study postulates that there were other triggers or states as well, which contributed to their formation. The existing COVID-19 vaccines targeted the spike proteins of the virus, which get attached to a healthy cell to produce the disease. An important direction from this study is that instead of attacking the spike protein, which are virus specific, that is, attached to a specific virus only, attacking the replication transcription complex, which being similar to a variety of viruses will open up possibility of same vaccine to be used for different types of viruses that produce the same type of replication transcription complex, thereby fighting various viruses by the same vaccine. Dr. Dana Farber, immunologist at Columbia University in New York, feels that this study lacks conclusive evidence about the role of T cells in this case. Dr. Malamini of University College of London, a co-author of the study acknowledging this, asserts that it cannot be a mere coincidence that 19 of the study group showed a greatly higher number of T cells that cleared infection in them. She feels that a human challenge trial in which the participants are deliberately exposed to SARS-CoV-2 would help to establish beyond doubt that the T cells are actually helping to clear the infection. Thank you for all listening. Please subscribe, like and share my video. Viewing once again will make you understand even better. See you very soon with another easy to understand and to like scientific and research work. Bye dear friends. Take care till we meet again and beyond. Your comments and suggestions are highly appreciated. Thanks again.